structure. So today we'll be discussing about the role of proteins in the membrane. And when we talk about proteins, you need to know about two, that is integral proteins and peripheral proteins. So the first thing we're going to do is try to actually understand what integral means and what peripheral means. So this is going to be confusing because the textbooks are not giving it properly. People are not teaching this also properly because I don't know, maybe they're not reading enough. So I read through a lot of articles, a lot of books. So I'm just going to put it out to you, okay, so that you make sense of this. And when you talk about integral means it is very intimately related to the phospholipid and the lipid structures in your membrane. So in your membrane there's a hydrophilic and a hydrophobic part. Okay, so this thing is intimately related to your phospholipid and covalently linked to your phospholipid. Okay, so when it's covalently linked, it's basically, there inside the hydrophobic part, in the core of it. So it is integrated and it is a very integral part of the membrane. And it's not easy to remove this. You put a substance like detergent to kind of, split the membrane. That is the only way you can actually remove the integral proteins. Okay, so this is very integral, integrated into the membrane. Okay, and is covalently linked. So that is the integral proteins. On the other hand, the peripheral proteins are there peripherally. Okay, they are not embedded into the membrane like your integral protein. They are not in line with the hydrophobic part of the membrane. They are peripheral. Peripheral, don't get confused. It's not only the extrinsic outside. It's there inside also. So outside the membrane, it can be, your peripheral proteins can be there in the extracellular layer or the cytosolic part. Remember this. So peripheral proteins can be the peripheral, but it is there either in the extracellular fluid or the intracellular fluid. So that is a peripheral protein, and that is not having any covalent attachments, non-covalently linked. So there can be ionic linkages or hydrogen bonds. So for disrupting them, you just have to do stuff like increase the salt concentration, decrease the salt concentration. That by itself can remove these substances out. Not like integral proteins which need a detergent. Okay, so this is the basic difference. So when you talk about integral proteins, there are different types. When you talk about integral, it can span the entire membrane, one side of the membrane to the other side. Transmembrane. This transmembrane can be having one domain, it can be having multiple. It can span the membrane multiple times. Apart from this can also be embedded partly in one leaflet and all that. So this is where people get it wrong. People generally tend to think that integral means yes, transmembrane, so it is integral. That is not how it works. Covalently linked, that is the important thing. And even with one leaflet, it's fine. Okay, so there can be transmembrane, there can be monotopic ones which are basically embedded into one leaflet. Next comes another peculiar type. Okay, this again might be a little confusing. So again, I'm trying to clear it out for you. So this type of molecules, these proteins, they are there outside. They are not there inside, as in the membrane. Okay, they are there outside actually. So you would mistake it for a peripheral protein, but they are linked to structures in the phospholipid through covalent linkages. So like GPI anchoring. So you take a phospholipid, say phosphoinositol, a glycosylated part of the phosphoinositol, to that this protein is attached covalently. So it is covalently linked, but it is there peripherally. So that again becomes a little confusing. Now there are other proteins which are lipidated to the fatty acid chain of the phospholipid. Okay, so that again confuses people a bit. So often a term called membrane associated proteins are used for these. Okay, now in case there is a doubt with peripheral and integral for these types, I'd be more inclined to go for integral proteins, because different textbooks still give different versions of this, but since it is covalently linked to some part of the lipid, I'd say it's best to consider this integral proteins. Okay, I'm telling this based on things I read. Okay, so that is it. So there is transmembrane, there is monotopic, and there is these other proteins, membrane associated proteins. Okay, then we have peripheral. Now once this is clear and this difference between them is clear, we'll try to understand what are the functions of your integral proteins. So let's talk about receptor and signaling and all that. Okay, so when this membrane spanning protein that we just saw, transmembrane protein, okay, you must have come across it many times in physiology. So this is a very common thing that happens. So this acts as a receptor. So this thing, okay, this protein structure which has an amino terminal as well as COH part, this one terminal amino part is where the ligand binds to it. That results in some conformational change that gets carried over and activates something else inside. So this acts as a receptor. Okay, a receptor for understanding what is happening outside and activating things inside. So this also helps in signaling ultimately. Apart from that GPA anchored thing and the lipidated ones that we were talking about earlier, they themselves directly are involved in signaling. Okay, so ultimately these integral proteins are involved in signaling mechanisms. So that is it. Next is adhesion. You see, the cells in your body are not just freely floating and moving across here and there. They're all, attached to each other and to the matrix. So for one cell to attach to another cell, you need things like adhering junction, desmosomes, okay, to maintain the paracellular flow. You have something called tight junctions and all that. Okay, so we just talk about adhering junction now. So for adhering junction, there's an integral protein called the cadherin. So this cadherin helps interact one cell to another. Okay. Similarly, to understand how it is interacting with your matrix, you need some substance called the integrin, which is an integral protein. So this integrin attaches itself to the fibronectin, laminin, collagen, all this extracellular things in the matrix. Okay, so that again helps it attach and also helps in signaling. All these also help in signaling partly. Okay, so that is how adhesion is also there. So these act as cell adhesion molecules. Now the most important thing, transport. We were talking about things that are ionic, hydrophilic, water soluble, or water that needs to pass through the membrane, but it has a hydrophobic core. So how are things going to pass through it? So for that, you have your protein structure that forms pores, channels, carriers, pumps, whatever. Okay, so how does this happen? You have to understand your protein is an amphiphilic structure. It has a hydrophilic part and a hydrophobic part. So this amphiphilic structure is a helical structure. So in this helix, this is a protein structure which has amino acids in it. So every 3 to 4 amino acid, a hydrophilic is replaced by a hydrophobic. It alternates. So every turn, if there is a hydrophilic, like hydrophilic, hydrophobic, hydrophilic, hydrophobic. So one side of that protein is going to be hydrophilic, the other side is going to be hydrophobic. Okay, so like that it can interact with the membrane, hydrophobic part like that, and this part can be hydrophilic. You put two things like this, you form a pore here, which is hydrophilic and will allow passage of hydrophilic substances. Understood? Yeah, so this is the thing. So every 3 to 4 means on an average 3.6 amino acid. Okay, so this amphiphilic structure has both hydrophilic and hydrophobic part, and this structure has one part hydrophilic, one part hydrophobic. Okay, so the hydrophobic part helps it get attached to the core of the membrane, while the hydrophilic part allows passage of substances, forming a pore. Now these things can be a pore, channel. These are very simple structures. simple diffusion. Okay, channels especially are gated. Pores is the opening that is slit. So, people use this term pore and channel, kind of in a similar way. But pore is that opening basically, and a gated version of that is your channel. These ultimately allow simple diffusion. You open it, based on the concentration difference, things go from high concentration to low concentration. Next is your carriers. Carriers help in facilitated diffusion. On one side they take it up, flip, conformational changes happen, take it to the other side. So with the help of this, they move stuff to one side and to the other side. This is facilitated diffusion. Then we have pumps. Pumps are sort of like enzymes that use energy. You split ATP, use the energy to move something against concentration gradient. So we have pore, channel, carrier, pumps, and I hope by now you know the difference between them and the basic structure. Now you see how wonderful all these things is. Nobody teaches you any of this because of course it's not important for your exams. But the clarity you get by understanding all this, elevates everything. Your whole studies, everything, it gets, you have a completely different picture. Every time you think of ion or a pump, you start seeing it all differently. Your membrane, for that simple reason. Okay, so that is done. Next is enzyme. So these integral proteins also act as enzymes. One example that I just talked about is your pump. Pump is actually an enzyme. Apart from that, just forget about that. Okay, let's take a look at your GIT. The final steps of digestion in the GIT needs in the intestine, the cells that are there, they have some integral proteins which are, enzymes, and this helps in the final digestion steps. Okay, so that again, these are hydrolysis. Okay, so that again is another important thing. So these enzymes, if they get removed by high and low salt concentration, what's going to happen? That's not feasible. So of course it's not a peripheral protein, that has to be an integral protein. Okay, so that is it about your integral proteins. Next we'll come to your peripheral proteins. So peripheral proteins are located outside or inside, important for signaling. Yeah, so we had talked about an integral protein, a transmembrane protein. Let's take an example of a metabotropic protein. Okay, receptor, metabotropic receptor. Now once that is activated, it's going to activate another peripheral protein which is attached to it, which is the G protein. Okay, so this G protein is actually nothing but a peripheral protein. Okay, it has 3 subunits and all that, but it's a peripheral protein. Now this is now going to go and activate other stuff. So that is how it is involved in signaling mechanisms. So that is it. Now the membrane structure. Okay, cell adhesion. So remember the cadherin that I talked about. So those adhering junctions, the cadherin that is there, that is attached to the cytoskeletal structures. To attach itself to cytoskeletal structure, it needs an intermediary peripheral protein, and that is your catenin. So cadherin is attached to catenin, and catenin is a peripheral protein. Now let's take a look at other submembranous cytoskeletal structures. Take a look at RBC for example. RBC has something called spectrin. It's a submembranous cytoskeletal structure, and this again is attached to actin. Okay, but the spectrin now should attach itself to an integral protein in the membrane called the band 3. For that, it needs a peripheral protein called the ankyrin. So ankyrin again, what it does is just anchors it to an integral protein. Okay, so this is where you have to understand integral proteins are there and peripheral proteins are there, and peripheral proteins can, attach itself non-covalently to your integral proteins as well. Okay, so that is done. And apart from that, some of these peripheral proteins, whether outside or inside, also have enzymatic activity. Okay, so that is it. Yeah, so let's just take a look at phospholipase C, enzymes like that. So these are all peripheral proteins. Okay, so I hope I was able to help you understand the importance of these proteins in the membrane and how they act. Okay, because understanding basic concept is the key. You understand basic concept, you understand physiology, you understand medicine, everything. Okay, so I hope this was clear and it was helpful. Thank you.